Swayam Prabha. Digital India. Educated India.
Good morning or good evening, depending on wherever you are. I am Professor Shantanu Bhattacharya, and I am an associate professor at the Department of Mechanical Engineering at the Indian Institute of Technology, Kanpur. Today I will be actually teaching you about this very fantastic area of microsystems fabrication by using advanced manufacturing process. So to begin with, I would just like to say a few lines about what really microsystems are, or how this microsystems technology has emerged over the years. So if you look at this particular slide, it kind of gives you a historical perspective of microsystems research and the way it evolved. And here especially this highlighted region, as you can see, talks about this very famous lecture by Sir Richard Feynman called "There is Plenty of Room at the Bottom." Okay, so it all started really from this particular lecture, which incidentally was presented at the annual meeting of the American Physical Society way far back in 1959. So at this time, really the research in the microsystems engineering area was fledgling, or at its fledgling stage, and there were some very interesting ideas which were pointed out by Dr. Feynman about what can happen really when one can go smaller and smaller. For example, in one instance he really pointed out that if you can write the whole Lord's Prayer on the tip of a simple ballpoint tip or ballpoint pen, how small can the letters be? And so he talked at length about different scales and how they would look at when you really do miniaturize them, and some of the very fundamental concepts like scanning electron microscope, transmission electron microscope, etc., and the power of high energy beams have really emerged from this lecture. So this really followed one of the most rapid technological developments, which is also known as the miniaturization of electronics and microelectronics, as we call nowadays. And there was this very famous law called Moore's law, which came into being from a business perspective, where it was pointed out that this miniaturization density, if you look at every 18 months, for a period of 18 months, would be essentially doubling, which means that the amount of circuits that you can really print onto a small square area would go twice exactly with the passage of about 18 months or so. So this law really also tells you about how small the miniaturization drive of electronics can take you to. It kind of gives you a time scale estimate for that. So this law, when it started, looked very good, and people really had about 18 months time period for approximately doubling of the integration density. However, because of the limitations posed by photolithography processes, I am going to describe this in details a little bit later, what photolithography means really. So because of the limitations posed by this photolithographic process of being able to go more than 100 nanometers or so, this law may eventually change to doubling in every 24 months. And nowadays increasingly it has been seen that instead of the 18-month period, the period of integration density doubling itself has shifted to about 24 months or so. So this is a sort of brief overview of where the microsystems drive or microsystems revolution really started from. So because of these limitations posed by the Moore's law to the business of the microelectronic industry, there were some processes really which fell out because they were no longer capable to handle the integration density which was offered by microelectronics. So in the late 70s, these technologies were kind of gathered together, and the silicon technology was extended to what you call machined mechanical micro devices, or MEMS, as the term is known, micro electromechanical systems, which came into being because of the fallout of the processes from the microelectronic industry. Okay. So, this field really was very exciting because it had a component of mechanical motion or mechanical synthesis of small miniaturized structures or features. It also had a component of electronics, where whatever signals it would be able to process can be transduced into corresponding electronic signals, and then of course it was at the micro scale. So, thus the name micro electromechanical systems. So, as this field emerged, microsystems technology increasingly got applied to many fields like fluidic and optical industry or components, and these sort of synergized with the MEMS technology and got integrated into this whole micro devices. The concept of devices actually came up, where variety of techniques like fluidics, optical components, microelectronic systems, micromechanical systems could be all integrated into one platform, and together these so-called micro devices were, and the way that it was realized or built, were known as the microsystems technology. So, our course really would focus more on this microsystems technology and some of the very state of the art processes of fabrication which should be used for realizing these systems, as we would see in details later on. So, let us look at a little more of historical perspective as to how the MEMS or micro electromechanical systems really got integrated into biological or biomedical MEMS. So, really when the MEMS started in the 1970s, it was dedicated mostly to physical systems like realization of temperature, or measurement of temperature, acceleration, velocity, so on and so forth. But towards the late 1980s, there were increasing amount of usages of this microsystems technology to realize micro flow sensors, micro pumps, micro valves, where fluidic motion at the micro scale suddenly became very important. I would also like to point out to this one famous paper by Sir Andreas Mans, which was presented at a plenary lecture on miniaturized total chemical analysis system, a novel concept for chemical sensing, and this was presented at the International Conference on Sensors and Actuators, which really changed the dimension for these micro electromechanical systems, and slowly and increasingly the field emerged into the biomedical micro devices or biomedical MEMS systems. So, what these bio-MEMS devices are, or what they do really, they are having relatively very high applicability to the field of life science, biotechnology and medicine, and of course, they are systems made up of very, very small micro to nanometer size components, and this is because primarily these MEMS features scale very well with some of the biological entities which are available. So, therefore, this concept of MEMS, which emerged from physical MEMS and went into biomedical MEMS, really has taken a lot of steps and research has emerged in this particular area. So, this really shows why MEMS can be applied to the biological world, and this is one slide which mentions something about the sizes and the scales and the comparisons of the various MEMS devices with respect to the biological entities. So, here if you look really at towards this left side of the scale, it starts from about 0.1 nanometers, as you can see, and it can go all the way to about 100 microns or so. And just to give you a few examples on the size domain, 100 microns typically is the size of a human hair, and these entities here, as you can see, are the biological entities which are very commonly available in different forms of life in the nature. For example, most plants and animal cells, if you really look at the size domain, are about 10 micron plus region. This is an illustration of a red blood cell. It's like a inverted button from a shirt. It is a small sac which contains various chemicals and is a very good oxygen transporter within the human system. This, for example, is E. coli. It is a small, cylindrical 2 to 3 micron length, about 1 or 1.5 microns diameter object, and this is a bacteria, and most of these bacteria are in the size range of about 1 micron to 10 microns. And if you look at this particular species here, it is a virus. It is a capsid made up of proteins with enclosing few genetic information or materials, and most of the viruses are in the 100 plus nanometer domain size range. If you go one step further, this, for example, is a protein molecule, and this here, as you can illustrate here, is a DNA, deoxyribonucleic acid, and one helical turn of the DNA is about 2 to 3 nanometers, 20-30 angstroms. So you can think of that Mother Nature kind of builds these systems up using a bottom-up technology, assembling atoms and molecules in a particular fashion, so that you can generate these smaller entities, biological entities, and these together would realize one of the very highest and most complicated forms of life, using bottom-up fabrication technique. On the other side, on the right side, you can see some of these structures and features from the common microsystems domain. This, for example, is a micro cantilever. You can see this lip sticking out of this structure, and this whole pool here, as you can see, is a sort of cavity into which this lip is sticking out. So you can recall this as a diving board in a swimming pool kind of structure. The only difference here is that because it is micro scale, this lip is about close to about tens of microns, and the thickness could be about 200 to 300 nanometers, and the depth into which this cavity is placed can be close to about 1 to 2 microns. So the reason why I am illustrating all this is that some of these features and structures rhyme very well size wise with these biological entities. For example, this is again another very interesting example. It's a polysilicon gate with nitride spacing, and the minimum feature size of a MOS transistor, which has been declared by the ITRS roadmap in the year 2004, is less than 100 nanometers. And for such a 100 nanometer device, the gate insulator region, which may be this region, is as low as a few nanometers, about 2-3 nanometers or so. And so you can see that even at the molecular level, there are these features and structures which are created, which rhyme very well size wise with these molecules. So it gives you an opportunity to sort of interact with the biological world more appropriately by building micro size features and objects, and therefore the MEMS changed its direction mostly into biomedical MEMS, and it had immense utility on some of the very interesting projects developed by the human civilization, like the Human Genome Project, so on and so forth. And so there was a huge class of materials called integrated biochips, which got formulated, and this is one of the major areas of MEMS, where some of these components could be microfluidics, or could be with molecular specific sensors, could be MEMS and NEMS systems, and molecular devices and memory structures, etc. They got integrated into these 2D CMOS platforms, and they could do integrated sensing, detection, diagnostics very rapidly. So therefore, I have kind of illustrated or walked you through the history of these microsystems technology, so called, started in 1970s as a fallout of the microelectronic revolution, and then basically went to realize some of the physical micro devices which could sense physical velocity, acceleration, pressure, so on and so forth, some physical parameters, and then finally emerged into this biological microsystems or biomedical micro devices which could do sensing, diagnostics very rapidly. So it has been a very useful area of research and work so far, and what we will learn in this course really is how to make some of these microsystems devices using conventional or most of the time advanced machining processes or machining technologies. Let us look at some of the examples of physical MEMS, as I told you before, and these are some of the commercially available MEMS structures and devices as on date in the industry. So, this mostly is an area which is realized using silicon, and let us see example 1 here. It is a bulk micro machined accelerometer, and this comes from a company called Silicon Microstructures, and typically this accelerometer is having a basic principle as illustrated here, the concept of a proof mass. So, if you look at this top left corner, we have schematically shown how this accelerometer works. So, this proof mass here is essentially a very small feature which can be about 20s of microns by 50s of microns, and this is pivoted typically. So, if you look at this domain cut, so if you look at this proof mass and the way it is pivoted, it is illustrated here by this small cartoon, okay, which I am making for you in the screen. So, essentially this mass is pivoted like this on a surface, and this surface here is actually is inside a cavity, or it is etched inside a cavity inside is etched, and the remaining part of the wafer is placed like this. Okay. So, you have a pivoted proof mass here by this shaded region, which is nothing as but this illustrated here, and you have a cavity here around and typically what happens is that there are metal structures which are coming out from this proof mass on both sides. Okay. So, these are some of the metal structures which are coming out on both sides and can be illustrated here as 1, this one, okay, this one 2, and this is again 3, this 4, okay, and similarly there are other 4 indicated by these arrows here on the right side. So, these metal structures are coming out, as you can see by the vertical shaded lines on both sides, and then there are certain other metal structures which are actually present on the top of this surface, which has been micro machined and out of which the cavity has been made, and these structures are present in an interdigitated manner, as you can see. So, you have one structure, this 1, okay, present between component 3 and component 4. So, it is sort of interdigitated and it is placed in that manner. So, whenever there is a movement of this particular MEMS housing, there is a change in the angle of this proof mass here. So, the proof mass may actually become something like this, okay, as you can illustrate, and can be illustrated in this diagram by the dotted line. And so, therefore, there is always a change in the interfacial area between these wings which are there on the mass, proof mass, and the metal structures which are printed on to the otherwise plane surface. And because of the change in the interfacial area, there is always a change in the interfacial capacitance. Okay. And because of this capacitance change, there is a signal, there is an electronic signal. So, if I can really calibrate this capacitance change with respect to the acceleration, roll, pitch, yaw, different physical parameters, that is really what an accelerometer measures. These accelerometers are increasingly used nowadays in automobile sector for airbag based actuation, and in fact very small accelerations or decelerations can be recorded very easily using some of these structures. The advantage is that this kind of an accelerometer would have a very minuscule least count, or the accuracy of measurement would be higher, because this proof mass may only be a few maybe about nanograms or so in weight. Okay. So, it is a very small amount of mass that you are really carving out, so that you can check physical parameters with great accuracy. Example 2 is this very interesting tool, which is also the cornerstone of nanotechnology. It is called an atomic force microscope, or AFM. You can see here is a silicon structure coming out, it is like cantilever, and the tip of this cantilever is atomically sharp. So, you can see this tip here, it is sharp to the size of a few atoms, and this again is used extensively for topological measurements of surfaces, where this cantilever tip is moved over a rough surface, and whatever crests and troughs are present on the surface are being monitored by means of the deflection, by monitoring the deflection in the z direction of this particular cantilever. So, therefore, if suppose I were to monitor these small valleys which are present over the surface, I would simply scan this cantilever on the top of these valleys, and wherever there is a valley, and if we have some positive pressure onto the top of the cantilever, it would go into the valley, and there would be a z direction displacement in the negative z direction. And if we can record that, we can really take all such data points and reconstruct the image of the surface based on some of these points. So, an AFM can really be used in the touching mode, or the tapping mode, and again silicon micro machining or micro manufacturing using some of these advanced manufacturing techniques are used for building some of these systems. So, along the course we might have an example problem where we might see how to develop or devise these AFM tips using non-conventional or advanced manufacturing. Example 3 here is a very interesting example. It is one of the commercially available devices which are available in the PowerPoint in the multimedia projectors, and this is something called a digital micromirror device chip from Texas Instruments. So, let us look at the basic principle of how this system works. So, you can see here in this particular figure an array of plates which are shiny, and they are probably made up of metal, and they are very teeny tiny. So, therefore, it is actually a blown up image, and each of these plates may be something like about 6 micron into 6 micron. Okay. So, here this dimension may be about 6 micrometers, and similarly this dimension may also be another 6 micrometers, and each of these plates are arranged in an array format in a very complex manner, as can be illustrated in this figure. So, these plates are really pivoted on both ends, and they are like a simply supported beam configuration, and these plates also are electrically actuated from the bottom structure, which is indicated in blue here. Okay, and they have these electrodes. So, if there is an electric field applied between this blue base and the bottom of this plate which is hanging, the plate deflects, and because of the deflection, if this plate was shiny on the surface, and if suppose light was falling onto this plate, light would go out of focus. So, here for example is the DMD chip, which actually is carved out of this array of different plates. And here for instance, if suppose this light beam falls in a certain direction from the focus, the optical focus, and there is some pre-processing done, like passing it through RGB filters and then optical choppers, etc. And let us say it were supposed to go in a direction towards the focus or the focusing lens. So, typically if the plate gets deflected, the beam would go out of focus, and so typically the pixel which was being represented by the plate which got deflected would close. So, you have a pixel close because of movement of the small mirror. Okay. So, you may be able to realize that the image which is projected is really a function of such pixels. It is an array of pixels. So, a pixel illustrated by a certain color or brightness or intensity would be representative of creating some impression on the surface which is corresponding to the image. Simultaneously, a pixel which is closed or a pixel which is switched off would represent a dark region of the particular image. So, it is really an array of these pixels which would be able to demonstrate an image, and you can do suitable amount of optical magnification etc. to this particular image to transmit it. And so, if you have a signal in form of a 0 or a 1, it can drive the angular deflection of several of these mirrors in unison, thus closing several pixels and opening several pixels, and that can be used to translate the digital information of an image that is recorded in a computer onto the projector. So, it is one of very fantastic examples of what MEMS structures can do in terms of image projection. Let us look at some of the other examples of MEMS. These are two very interesting examples of silicon MEMS. Again, an accelerometer device from Analog Devices, which is used typically for the deployment of airbags, as I was mentioning. And this is again a single chip microphone, which is again the principle is based on this vibrating membrane, which is highly perforated, as you can see on the bottom, and you have a layer of piezoelectric material coated on the top. So, whatever you are speaking into this small microphone basically resonates and vibrates this perforated membrane, and it creates an electrical signal which can be processed or magnified, and that is how you can hear sound transmitted electronically over distances. So, typically all speaker based systems like telephones, cell phones, microphones etc. are based on some of these miniaturized or super miniaturized principles at work. So, that is some of the examples of silicon MEMS. I would now like to also illustrate some other examples. So, let us now see some more illustrations on applications of MEMS or microsystems in biology. And here I have for you about 6 different illustrations of how microsystems can be really used in biology. And as you can see here, the first example is very widely referred to and read, is basically this set of microelectrodes which were developed at the University of Michigan by Dr. Wise's team actually. So, essentially this was used for monitoring the electrical activity of brain tissue, and these electrodes here, as you can see, are printed and they are in plane, and this needle here, needle like structure here, may be only a few tens of microns in width and a few hundreds of microns in length, and they are all printed with these so called electronically placed or for electrical identification of signals. They are all coated metal prints or imprints placed on the top of these electrodes. This again another is a very this is again another very fascinating example from research group at Stanford, and this is a neuroprobe, and it is used for providing deep electrical stimulation to patients suffering from Parkinson's disease, where these stimulations are medically advised sometimes for proper operations, or for the sort of routinizing of functionalities of any human being suffering from these diseases. So again, a very fantastic example of microsystems at work. Slender like tall needles, which may be about 20 to 30 microns and width wise, and few hundred microns tall, and these are quite excellent examples of microsystems in biology. This is another example of what microsystems can do for monitoring the crosstalk between neurons. These are some neuron cells growing over an array of MOSFETs, and this area is also known as nanobiology, and it has recently come out very well. Sometimes physiological monitoring of cells using this signals, these electronic signals, becomes very critical to understanding their crosstalks. So essentially the idea is that these neurons are growing over the gate region of these MOS transistor arrays, and if there is a change in the surface protein expression of these cells, it's recorded in terms of change of surface potential. And so if you can keep on monitoring the current based on the change in surface potential, it can give you an idea of what crosstalk is going on a single cell level between many cells growing together on the top of this array. Example 4 here is again a very interesting and very fascinating example of MEMS in biology. This is a micro needle, okay, and this is based on the sting or the needle of a mosquito. So when a mosquito bites you, you hardly feel the pain because the needle is not able to really damage or deflect the pain receptors which are placed underneath the human skin. So of course there is a swelling which comes, but that swelling is more because of an enzyme that it releases, so that it can thin out the blood samples that it is taking. But otherwise these needles are about 20 microns and again about 180 or 200 microns in length, 20 microns in diameter. So it's a high aspect ratio, slender structure, and such structures, if we can fabricate as arrays or groups and fabricate it over, let's say, pads which can be mounted on the fingers or different regions of the body, you can think of or envision a system of painless drug delivery to the human body. So in fact this slide has been borrowed, or this picture has been borrowed from this company called Cometrix, which commercialized some of these microneedles technologies for drug delivery applications. This again is an example of a very fascinating area of microsystems called integrated biochips and sensors. Now this area also got into picture because of the need dictated by the Human Genome Project again, where analysis of samples became very critical at a very rapid pace or fast pace. So typically these biochips, integrated biochips, are made up of some channels and chambers and architectures which can handle minuscule amount of volumes of fluids of a certain target, and they can be mixed in proportions, different proportions with their analytes of interest, so that they can be recognized. So there is some kind of a transduction of signal which changes the chemical information onto the analyte of interest into an electrical signal or an optical signal, and the whole integration of how to change the signal or how to process the signal or how to infer from the signal is done together on this integrated biochip. So these technologies, or this group of technologies, are also known as lab on chip kind of technologies, and they are very useful for the diagnostics, the clinical diagnostics world. Again, a very fascinating application of MEMS. Again, this example 6 here illustrates a bunch of different cantilevers, and I have already mentioned about these cantilevers extensively while talking about AFM and the technology of AFM. So, these cantilevers can be very easily used for recognizing DNA molecules. In fact, this work done for the first time at IBM Zurich Research indicated that deflection on these cantilevers can be synergistic with some of the binding events that may happen onto the top of these cantilevers between capture probes, which are very intelligently placed on the top of each cantilever, and a target molecule which may come and bind. And so this deflection can be recorded with certainty to indicate whether there is a binding action which has happened, and this can be used for typically for applications like DNA microarrays, etc. So, this is again another very fantastic example of microsystems in biology and its applications. So, if we really look at a bunch of these different technologies, sort of micro and nano systems, and the way that they apply to the the biological world. So, I would try to classify these technologies as ones which have a biological impact or are bio inspired, and it's a synergistic learning process where on the left side these three boxes are representative of learning experiences from micro nano systems which would result in some novel solutions for frontiers in medicine and biology, and these can use examples like diagnostics. We have discussed about biochips. There can be nano microstructures like quantum dots, silicon nanowires, carbon nanotubes, so on and so forth. There can be some therapeutic applications like targeted drug delivery, painless drug delivery or drug release systems, again where micro nano systems can be used for developing some solutions or frontiers in medicine and biology. And then you have these hybrid bio devices where you can develop through tissue engineering a complete organ, but again keeping the principles of micro nano fabrication or advanced manufacturing of such processes as applied to biology and medicine. Okay. The boxes on the right here are illustrative of a reverse learning experience which you get through what biology and medicine has to offer and how you can apply onto some systems which would be useful for developing micro nanotechnologies. For example, let us consider self assembly, which can be DNA protein mediated or mediated with SAMs or self assembled monolayers. Again, something which is naturally available, and you can directly apply this to do some very novel work of maybe nanoscale interconnects or microscale interconnects, which can be a part also of molecular electronics, where single molecules or DNA can be used as a basis of conducting wires, nanowires between more than two posts. There's another very fantastic example of developing these bio-inspired materials, like for example the human skin. Okay, so if you look at the skin, it's such a wonderful instrument. It can self-heal, it can respond, it can repair, it can sense, and learning from what Mother Nature has to offer in terms of this scheme and trying to enumerate the fabrication technology so that you can develop these advanced machining processes is another way of looking to develop these novel solutions or frontiers in materials or information processing. So therefore, it is a synergistic learning experience. On the left you have learning from micro nano systems as applied to the biological world, and on the right you have learning from biological world as applied to micro nanotechnology systems.